Hi, everyone. On behalf of Metabolon, I'd like to welcome you to Reflections of the Human Gut Microbiome in the Human Gut Microbiome in the Blood Microbiome. My name is Carolyn, and I'm the host and moderator for today's event. Before I introduce our presenters, I'd like to provide some brief instructions. The presentation has been muted, but we welcome questions that could be answered during the Q&A session at the end. To submit a question, please open the Q&A window at the bottom of your screen. We will also feature, no, we will not. Now I'd like to introduce our presenters for today. They are Sean Gibbons, Ph.D., Assistant Professor at the Institute for Systems Biology, and Greg Michelotti, Ph.D., Scientific Director of Biology at Metabolon. Welcome and thank you for being here. Greg, the presenter role is yours.
Thanks, Carolyn. I appreciate that introduction, and it's really exciting to be here today talking about the human microbiome and the blood metabolome and how Metabolon has worked with the Institute of Science Systems Biology to really provide some really novel insights into alpha diversity, gut diversity, and its role in human health and wellness. By way of background, I'd like to talk about some initial aspects of what metabolomics is and why metabolomics is so important for providing insights into biological systems. Metabolomics is unique in that it integrates both genetic and non-genetic information to provide a comprehensive view of a phenotype, which is exemplified here on this slide, showing the central dogma of molecular biology, which is kind of encapsulated under genome on the left part of the panel, as well as integrating both microbiome information and environmental information to give a biochemical readout of metabolites that provide a comprehensive view of a phenotype. And at Metabolon, we focus on the comprehensive and accurate profiling of biochemicals and biological systems to provide a comprehensive view of what this phenotype is. And I do like to point out that there's a level of complexity on the left part of that panel, the central dogma of molecular biology, of DNA to RNA and protein, that all these factors go through a bewildering array of modifications that really confound our ability to understand how they behave across different model systems, phylogenetically, and even within a species, within individuals. Unlike metabolomics that are structurally conserved from bacteria to humans, and that structural conservation implies a functional conservation. And again, unlike genes, metabolites are constantly changing and responding to the environment, so there's a constant need to reassess and reevaluate the biochemical profiles, so there's a need for continuous monitoring in that point to give insights into a phenotype, and in the case of humans, health and wellness in transition to disease. And within this context, I'm really excited to have Sean Gibbons here, as Carolyn introduced, Assistant Professor at the Institute of Systems Biology, as well as a distinguished investigator at the Washington Research Foundation, to talk about his recent work with us. So Sean and his colleagues have done a lot of work to utilize our metabolomics technology to help understand the role of metabolic perturbations in really the transitioning from health and wellness to disease. And today we're really excited to discuss the specific application of metabolomic profiling or biochemical profiling to understand the diversity of the gut microbiome from circulating metabolites, which is pretty exciting, and what the implications of that are for obesity and obesogenic disorders. So with that, Sean, I turn it over to you.
Thanks, Greg, and thanks, Carolyn, and thanks to Metabolon for providing me with the opportunity to talk to you all about work coming out of the ISB. Let's see if I can share my screen here. Okay. All right. So, yeah, I'm going to give a little bit of background. This will be a very brief set of slides on the microbiome, and then go over some of the major findings of our paper and kind of skim over the top, and then save a lot of our time for Q&A after that. So, like was said, right, the metabolic capacity of our bodies, the metabolic phenotype of the human being is shaped by a lot of different factors. For a long time, we thought of these sort of organismal filters that shape our responses to environmental inputs, the sort of nature versus nurture. The nature side of the equation was the human genome. Right? You sequence the human genome and you're going to understand disease. You're going to understand, if you're exposed to this chemical, this is going to be the type of cancer you might be susceptible to given you have this mutation. And we've learned a bunch about that over the past few decades since the Human Genome Project, but it's become clear that it's a much more complicated story than that, right? There's a lot more signal in our bodies that beyond our genomes that are acting as these organismal filters that shape how our bodies respond in it going into either health or disease phenotypes given particular environmental inputs. And the microbiome has emerged as a really important component of this. So for those who aren't familiar with the microbiome, it's simply the microorganisms that live in and on the human body. Vast majority of these organisms are living in the colon, in the human gut. About 99% of all the biomass in our bodies that's microbial is in the gut. There's about as many bacterial cells in and on our bodies as there are human cells, a few trillion. Depending on where your last bowel movement was, you might be more human than microbe. And when you look at the genetic component of our bodies, there's about 100 times more genes in our microbiome than in the human genome. It's about 23,000 genes in the human genome and maybe 2 to 3 million genes in the microbiome. And a lot of those genes are important for shaping our phenotype and for how we respond to the environment. A lot of those genes aren't important for that. They're important to the microbes and maybe not important to us. And we're still just beginning to understand how the microbiome shapes the human phenotypes. There's a lot that's unknown. There's a lot of smoke, right? There's a lot of associations between health and disease for what species are upregulated or enriched in a given disease condition. But there's not as much known about how the microbiome contributes to the etiology of disease. Okay, so I think I'll stop pontificating here, other than to say that another unique component of the microbiome is it's like an organ of the body, right? It contributes to a lot of the metabolism of our bodies, but it's a self-assembled organism. We're born sterile, and we actually recruit our microbiota from the environment as we develop. Some of our strains stick with us from childhood, and we die with them, right? Perhaps. Decades of time, these organisms can stay in our system. There's also a lot of turnover where we can acquire new organisms, and there's just so much we have yet to understand about that process and how that shapes our bodies. So today, we're going to zoom into a particular piece of this puzzle with some work that recently came out of the Institute for Systems Biology, looking at the diversity of our gut microbiome. And this is a collaboration with two of my colleagues at the Institute, Lee Hood and Nathan Price, a couple of other faculty members. But the real heroes of this story are two really talented postdocs, Tomasz Wolmanski and Noah Rappaport. Tom and Noah did all the actual work to make this study happen. And so this was the beginnings of a strategy that my lab and Nathan's lab are very interested in, which is to see the imprint of the microbiome in the metabolism of the host organism or in the phenotype of the host organism, right? We know there's a lot of variation and fluctuation in the ecology and composition of the microbiome across humans, but how much of that variance is actually coherent with variance in the molecular phenotype of the host organism? And the first thing we wanted to look at was alpha diversity, because we had some clues that the actual diversity level, the species diversity, say how many species are there, or diversity in terms of dominance, right? Like how dominant is the most abundant species, or how equally distributed are individuals across species. This particular metric, there was good evidence to show that there was a coherence between this metric and the blood metabolome. There have been some prior papers showing that, for example, the metabolite hippurate, which circulates in the blood, was correlated with alpha diversity. So we thought this was an interesting thing to target, because as I'll discuss in a slide or two, there was also some interesting clinical implications for this particular gut microbiome metric. So we'll jump straight to the point here and say that we were able to actually see the ecological structure of the gut microbiome, some component of that structure, right? This diversity of the gut microbiome in circulating blood metabolites. By looking at some 1,000 metabolites that could be quantified in Metabolon's metabolite panel from the blood, we found that there was a sparse model of only 40 of these metabolites that could explain about half of the variation in the ecological diversity of the gut microbiome. Here we're plotting Shannon diversity, which is a sort of mixture of sort of dominance and number of species. But even if you look at just number of species or evenness, dominance by itself, the story remains the same. So, that's pretty surprising. We can actually see something about the gut without measuring poop, right? We can see it through the blood. And if you ask which metabolites are these that we're seeing as predictive of the composition and structure of the microbiome, most of them were lipids, although most of the panel was also lipids. Most of the things we could quantify were lipids. But we saw some, dipeptides, tripeptides, some steroid hormones, and other things as well. And if we kind of filter down that list of 40 metabolites to a set that were very, very consistent across different iterations of the model, looking at different validation cohorts and permutations, there was an 11 metabolite model that explained most of the variation in alpha diversity. And that's what you see on the far right of the screen. And these are the beta coefficients. These are coefficients from our lasso model, so essentially linear regression coefficients from multilinear regression. Positive means you're positively correlated with diversity. Negative means you're negatively correlated with diversity. And what we saw here was very satisfying in a sense, because it was a sanity check. A lot of the metabolites that we were seeing were microbe host co-metabolites. Now, these are metabolites that are only in the blood because of the activity of our microbiota. And those are what are labeled in yellow. So hippurate and cinnamoglycine, these are essentially degradation products of complex plant polysaccharides that are absorbed across the wall of our colon into the bloodstream, travel to the liver, and then are conjugated with a glycine, an amino acid, by our liver. So hippurate is a benzoate molecule that our microbiome has produced that we've absorbed and had a glycine added to it that makes it into hippurate. And these are both kind of markers of a healthy diet in a way, right? If you have a lot of this in circulation, you're probably eating a lot of complex plant polysaccharides. P-creosyl sulfate is a bit of a different story. That's a protein fermentation byproduct. And it has an ambiguous relationship to health. In people who have issues with their kidneys, kidney damage, P-creosyl sulfate is actually quite toxic. However, in people who are healthy or have healthy weights or have perfectly functioning kidneys, it's also been associated with positive health outcomes. So it's a bit of an ambiguous picture there. But as our microbes ferment proteins in our colon, we see more of this accumulate in the bloodstream. And the other two yellows are bile acids. So we produce primary bile acids, the microbes chew on those primary bile acids to produce secondary bile acids, and then we see those reabsorbed back into circulation, and that's what we're detecting. The blacks are, as far as we can tell, host metabolites that are associated with gut microbiome diversity. The most explanatory single variable was this 5-alpha androsten 3-beta-17 alpha in the top right there. And this is a derivative of testosterone. So it's a sex hormone derivative. We saw a few of these come up in this list of 40 metabolites that were explanatory of diversity. And whether or not this is a purely host metabolite, or perhaps it's a derivative due to the fact that a microbe maybe chewed on a testosterone molecule and produced this derivative, that we don't know very well, but it is known that microbes do kind of modify and chew on sex hormones. So this is sort of a high level view of the fact that we can predict diversity in the gut microbiome, and what are those specific metabolites that seem to be most predictive of that. And so here's kind of going back to like a clinical perspective of why might we want to predict alpha diversity? What is the actual clinical relevance of it? Generally speaking, I don't know if there's a huge clinical relevance to diversity in and of itself within a normal range of diversity, but it is known that if you fall below a certain level of alpha diversity, it's sort of a signature of you not being very healthy, right? So oftentimes we find people with diarrhea, people who've taken a lot of antibiotics, or people who have abdominal pain. These folks tend to have a much reduced gut alpha diversity compared to everyone else. It's like, a forest fire has kind of gone through their gut ecosystem, and there's only a few species left, and it needs to be kind of recolonized and regrow back again. And this is some result from a paper that I worked on in my postdoc lab, in Eric Alm's lab, with great postdoc Sepe Deepak-Poor, who's now running her own lab in UBC, where she showed that if you looked at the pre-treatment gut microbiome diversity of patients with an initial C. diff infection, it was those patients that had a reduced alpha diversity at the beginning that were much more likely to have recurrent C. diff infections. So you could, to some extent, predict C. diff recurrence by that alpha diversity level. And so, part of our thinking here is if you can predict alpha diversity through the blood, this could be an interesting way to integrate microbiome science into the clinical world, because blood is sort of the window into clinical diagnostics. And to sort of push this idea home a little bit more, the cartoon on the left here is something I just sort of drew up by hand, but it reflects basically microbial ecologists' understanding of the gut microbiome, in that there is a normal range of alpha diversity in the gut microbiome. But if you start to drop beneath a threshold of alpha diversity, the sort of metabolic niche saturation starts to drop off, right? So normally our commensal microbiota, they protect us from invasive pathogens. And one of the major ways they protect us is that they actually just soak up and use up all the metabolic resources that normally would be those resources required for an invader to invade. They actually utilize enough of those resources so that it's impossible for that organism to gain traction and to actually enter into the system. But if you knock out critical functional components of the microbiome to the point where those resources are no longer consumed and become more available, those are these windows of opportunities where invasive enteric pathogens can get into the system. So this is why I'm showing the sort of relationship between invasibility and niche saturation and alpha diversity. If you drop below a threshold, you become likelier and likelier to be invaded by an opportunistic pathogen. And so if we can classify people who are below some alpha diversity threshold, well, then perhaps we can stratify those patients and put them in a high risk category for things like C. diff infections. And the proof of concept, this is what this on the right here is showing, is just an ROC curve of, if we just use that 11 metabolite model, so we're no longer using all 40 of those metabolites in our original model, we're just using a sparser model of 11 metabolites and predicting the lowest quartile of alpha diversity in two different populations: the population that we trained the model on, and a separate validation population that we did not train the model on. And we can show that, with an AUC of 0.88, you can sort of predict who has this lowest quartile of alpha diversity as a potential application of we can see these people through their blood metabolites, which ones have this maybe high risk phenotype. And with that, I'll end off so we can spend most of our time on question and answer. But again, I'm looping this back to this idea of, we're at ISB, we're very interested in what amount of the variation in the ecology of the microbiome is actually functionally impacting the physiology and phenotype of the host organism. We're trying to see the structure of the microbiome through its impact on the host. There's probably many, many ways to have a healthy microbiome. There's many combinations of functionally redundant species that you can have that's totally fine and totally healthy, that give the same sort of metabolic output. So maybe we should be looking at the metabolic output as the major factor to analyze, to ask the question, do you have a healthy microbiome or don't you? And this is just a cartoon that a really great artist at ISB made, Alison Kudla, that we suggested as cover art for the paper, for the journal, but it wasn't accepted, sadly, where you have these microbial constellations in the starry sky in the shape of the metabolites they're producing, and then the reflection of these sort of metabolites in the blood red ocean. And with that, I think I'll stop talking and hand it back over to Greg.
Great. Now, thank you, Sean. That was really very, very interesting. In terms of questions, Carolyn, I don't know if you've seen it yet that could be fed to me, but I'll start out with some questions, because great studies beget additional questions. So the focus of the discussion is on the gut microbiome, but what do you think the involvement is of other microbial communities, such as the oral microbiome, genitourinary microbiome, pulmonary, et cetera, that could influence circulating metabolites as well? So, the gut microbiome is the 800-pound gorilla, if you will, but certainly in at-risk populations, they're also going to have divergence in these other populations as well. What do you think their relative contributions are, and have you thought about ways to go about studying them as separate communities?
That's a great question. It is true that the gut is sort of the 800-pound gorilla, right? The 99% of the biomass, what have you, of the body, microbially speaking, is in the gut. So you might expect that a lot of the sort of functionality is pent up there. But, for specific disease conditions, for specific applications, the microbiota living in different body sites are super important, right? Like the oral microbiome is obviously quite important to cavities, to the formation of cavities, to periodontitis. And so studying those interactions between microbiota and these different oral pathologies is super interesting and super important. There's also a bunch of risk factors for oral microbes that can, cause organ damage, like heart damage. And there's a lot of interesting evidence showing that in conditions like colorectal cancer or other diseases, you find that there's an enrichment for oral taxa on these, say, polyps, for example, on colon polyps. You see like Fusobacterium and a few other oral organisms that you normally see in the mouth becoming enriched in the gut. And so it is the mouth is sort of a portal into the rest of the gut system. And there's a selection to pass into the colon for organisms that are adapted to live there. But perhaps if the system's perturbed, if there's something wrong, some of those opportunists get in and start causing problems. This is I remember there was a Nature Medicine paper a while back about nosocomial infections and how, each one of us is kind of carrying our own, cohort of opportunistic pathogens with us into the hospital, right? It's not like you can completely obliterate nosocomial infections by sterilizing everything inside of a hospital, because we're all carrying it in with us. So we have the harbingers of our own doom, like living on our bodies. It's only the fact that we are healthy and we have a healthy microbiota that prevent us from getting sick from those things. And then final example is, the vaginal microbiome. That's a really interesting system to study. It's very different from the gut. It's a very dominated system by lactobacilli. And it's super important for things like preterm birth. And the metabolites produced by dysbiotic vaginal microbiota could have a huge impact there. So depending on the disease condition, yes, these other environments are very important. And all you need to do to investigate them is to just get samples from those environments.
Yeah, great. It would be really, really interesting to do that, I think. We have a number of questions coming in from the outside. So we'll just start it off with the top here. How sensitive is metabolomics compared to measuring actual fecal microbiome? And I think what they may be getting at is comparing the results from circulating metabolites, how relevant that may be clinically, or what kind of differential insights may be provided from comparing fecal metabolomics and microbiome activity to circulating metabolites. Do you have a sense for the different information that those two matrices might impart? Do you need both? Or could circulating be sufficient given everything that you said about, being the key to diagnostics potentially?
Right. I mean, it's a very complicated question. And I think maybe part of what that question is getting at is, can I just measure the metabolome and not need to measure the poop to get an accurate reflection of the microbiome? Yeah. I mean, I don't think that's the case at all right now, right? So we could only explain about half of the variation in alpha diversity looking through the blood metabolome. So it's still a pretty noisy, it's like you smearing some Vaseline on the window to look in at the microbiome. It's not a clear picture. It's a blurred reflection in the blood. And so, yeah, you're going to get a much more accurate quantification of the composition and structure of the microbiome by sampling the poop itself. However, the interesting thing about the blood is you're seeing what's coherent with those molecules that are bathing all of the organs of the human body, right? So this is like, even though it's a blurred reflection, you're seeing what component of the variation in the microbiota is actually driving something inside of the host. So perhaps whatever this, even though it's a noisy reflection, perhaps what variance is explained is the more important component of the variance when it comes to defining health and wellness from the human side.
Very cool. Another question. Do you think that disease states such as increasing intestinal permeability would affect the predictability of these blood biomarkers on gut diversity?
Great question. And actually this sort of allows me to bring in another piece of information from the paper that I didn't touch upon, which was the relationship between obesity and this prediction, this alpha diversity prediction. So, what we found is, this predictive quality of the blood metabolome for alpha diversity was quite robust. No matter how you sliced and diced the data or looked at different validation cohorts that you didn't initially train on, the predictions were very robust, except for if we looked at people that had a very high BMI. So people who were very obese, say BMI of higher than 35, the model began to fall apart. We could no longer use this 40 metabolite model to predict alpha diversity. So something about this sort of state of the system seems to have shifted. The metabolic interplay between the microbes and the blood that was happening in most folks starts to either change to a different state or kind of fall apart in extreme obesity. And we do know that in very obese people, you have increased inflammation, you likely have increased gut permeability. And so there's a more leakiness of like stuff getting across that gut blood barrier that maybe would be better filtered in people that weren't extremely obese. And so that was interesting. And in addition, we didn't have enough data really to train a new model on just those obese folks, but we did notice that certain metabolites became less predictive. So for example, the sex steroid derivative that we found is the most predictive single analyte for alpha diversity in most other folks, that lost its correlation with alpha diversity in severe obesity. But there was another metabolite, this PFOS, which is, I believe in like Scotchgard. It's one of these chemicals that bioaccumulates in the body over time. That was actually correlated with alpha diversity in severely obese people much more strongly than it was in the rest of the population. And a possible explanation there is that adipose tissue is better at bioaccumulating this particular compound. So perhaps you would see an enrichment of it in people who are overweight. But long answer to the question, yes, changes in permeability, changes in that relationship between the blood and the microbiome can disrupt our prediction.
And speaking of prediction, another question is, are you considering studies to see what metabolites predict beta diversity?
Great question. Yeah, so alpha, beta diversity, these are all these derived metrics that sort of abstract away some of the, complexity of the microbiome in the sense of, what particular species are in what particular individual. So generally, human to human, you find about maybe 30% overlap between two people in the species that they share in their gut microbiomes. And then, 3 people, it drops to like, 7-8%, and then it keeps dropping as you add more people. So everyone has a very unique kind of ecological composition of their microbiomes. And yet most people have a functional set, right? It's a metabolically complete picture. So there's lots of ways to build this healthy microbiome. So there's lots of this beta diversity, right? The difference between ecosystems across people. We are looking at beta diversity as well, and how that's reflected in the blood metabolome. There's another paper or preprint right now that Tom Womansky was spearheaded on aging in the microbiome that went on bioRxiv month and a half ago. And in that particular paper, we actually find an interesting association between a beta diversity metric that we're calling uniqueness, which is how far away are you from the nearest person in beta diversity space? Like how unique does your microbiome look? It appears that we become more and more unique as we get older, but only if we're aging healthily. So people who are on a healthy aging trajectory, that you see a sort of reduction in the core microbes in their guts, that the kind of core taxa that tend to be shared across people, like Bacteroides, Faecalibacterium prausnitzii, and an increase in the dominance of these less core taxa as you get older and older and older. And there is an interesting signature in the blood metabolome. So we see that this uniqueness feature is actually associated with markers of healthy aging that have been previously identified. So phenylacetyglutamine is one of them. It's been shown in centenarians to be enriched in their bloodstreams. And then we see associations with things like indoles, which are tryptophan derivatives, and have been associated with lower inflammation, and a few other compounds like secondary bile acids and such. So yes, we are looking at beta diversity in the metabolome as well.
Oh, very cool. Carolyn, I'm sorry that I don't have an idea of the time here. What is our timing on everything?
We have about half an hour more as long as you got
Oh,
it.
perfect. Okay, good. I thought we were running out of an hour, because there are many more questions that have come in. So in terms of the best matrices to go after, I think there's a constellation of questions surrounding that, because we've focused on circulating metabolites. But have you considered other matrices or looked at other matrices, Sean, in terms of urine or other things that may be providing a more complete picture, a distinct picture? Do you need both? What are your thoughts on that?
That's a great question. We haven't looked at urine in my lab. Perhaps I think there are others at ISB who have looked at urinary metabolites. One other matrix we have been looking at is feces. So looking at the actual metabolites in the feces, there's a lot we still don't understand about the metabolites that are produced in the gut. Of those, how many are turning over and being broken down and metabolized by other microbes versus how many are being absorbed by the host, and how many of those that are absorbed by the host are being broken down very rapidly, and how many of those are going into circulation and kind of sticking around long enough to be able to measure them. And so a lot of this sort of absorption and kinetics of consumption and transformation is very much not understood. A good example here are short chain fatty acids. So we know, most of the complex polysaccharides in our diets are fermented down into these SCFAs, or short chain fatty acids, like butyrate, acetate, propionate. And it's known that these are very important to health, right? Like butyrate is an important energy source for our colonocytes. It also has been shown to be anti-inflammatory. It upregulates T regulatory cells. Propionate has also similarly been shown to be anti-inflammatory. And be involved in like glucose metabolism in the liver. But, it's been a few studies that have measured these SCFA levels in stool and found that there's like not much coherence between sort of the raw stool sample SCFA profile and the gut microbiome. So that's interesting, right? There should be a good coherence there, and yet it's not so great. And so part of the reason we think that is that the host is absorbing and breaking these down so quickly that whatever profile you measure in the feces is perhaps not very reflective of the in vivo production of those particular metabolites. So in our group, we've been doing these experiments called ex vivo incubation assays, where you take someone's stool and you make a homogenate out of it and bring it into the anaerobic chamber at 37 degrees C body temperature. And you can add a fiber like inulin or pectin or what have you to that stool sample and incubate it for a few hours, and then actually measure a couple of time points and look at the fluxes of metabolites that are produced or consumed over that time window. And these ex vivo flux measurements actually show a much better correspondence to the microbiome structure. There's a recent preprint from my postdoc lab, Eric Alm's group, showing this, than the sort of raw in vivo measure of like what SCFA is just sitting there in stool. It seems that you might actually have to measure the production flux of the living microbes that are in that stool in order to get a good view of that. So, I mean, that was a very long-winded answer to the question, but yes, we're looking at other matrices, and the more we can incorporate these other models of how things are produced, how they're consumed, where they're flowing throughout the body, the better we'll be able to kind of predict and understand the system.
Yeah, and I don't think there are any short answers to these as we realize the gaps in knowledge here and the studies, but this helps inform the studies that need to be done to help fill in these gaps in knowledge and provide a comprehensive understanding of the interplay between host and microbiome. Within this context, there are a number of questions regarding specific contribution of GI structures to circulating metabolites and how that may inform disease. So for example, some questions about bowel resections and how that informs circulating metabolites and health and wellness subsequent to the resection, and or are there studies that have been done, or do you know if you can comment on specific structures of the GI tract, ileum, duodenum, jejunum, et cetera, and going down to transverse colon, descending colon? Are those structures imparting differential changes in metabolomic profiling and how that may inform health and wellness and transition to disease? Is that known? Are you working on anything like that?
We had an R1 proposal with Nathan a year ago to look at stomach stapling surgeries and how that affected, say, metabolite profiles and
Yeah.
microbiome. Sadly, that didn't get funded, but I know there are a lot of folks that are working on those types of questions. We've been working on stuff from a modeling standpoint. So Christian Diener in my group has been doing a lot of metabolic modeling of the gut microbiome. Mostly we focus on the sort of conditions we expect to see in the colon, because that's actually where most of the activity and metabolism of our microbes is occurring. Although you do see, there's definitely stuff happening higher up in like the small intestine or at the sort of interface between the small intestine and the colon, where, the bile ducts are, and you have the primary bile acids being incorporated in. Not a ton of data on humans for that sort of thing. There are a few labs that have done resection experiments in animal models, in mice. Peter Dornstein just had an interesting paper out of UCSD looking at germ-free versus mice with microbiomes, different organs of their bodies and what metabolites are there and what aren't. I've seen some data from macaque monkeys where there have been some dissections of different parts of the gastrointestinal tract and what bugs are there, how different is the small intestine from the upper colon, and so on. And there's a few papers I've seen in review showing metabolite profiles as you move down the gastrointestinal tract. But we're in very early days, I would say, of just beginning to accumulate that data and just do the basic natural history necessary to have a map to understand that terrain.
Yeah, so within that context, are studies being done in terms of, because you mentioned, so plant polysaccharides and how that's informing potential functional outputs from specific bacterial taxa. The correlation between components of diet and the microbial community, the diversity or complexity of the microbial community. So question comes in, is it possible that the microbial community itself may not change, but the functional output changes depending on the dietary input?
That's a great question. I mean, this is part of what we're trying to get at with metabolic modeling. So Christian and my group, essentially, you could have me as an individual, my diet changes fairly drastically from day to day, or it can, right? Like one day I'll have an artichoke and some beans, and the next day I'll eat a steak. My microbiome is staying fairly stable in that time frame, and yet the metabolites, the sort of metabolic capacity of who's doing what and what metabolites are produced could vary drastically. And we know this just from modeling in and of itself. We can build these flux balance analysis models of a person's microbiome based on the sort of genomes that are there and the biochemical capacity that we understand about the system. And you can feed in different dietary stoichiometries. And, if you feed in, the combinatorics of different compounds actually affect the outcome of what's produced. So yes, the short answer is yes, you can get different metabolic outputs for the same microbiome depending on the inputs. And if you want to flip that on its head, you can input the same exact metabolite into two different people. And because they have different microbiota, the metabolic outputs are also different. And so we've shown this in some preliminary data we've gotten for a grant, where if you do these ex vivo stool incubation assays to measure short chain fatty acids, for example, you have person A and person B. This person has more Firmicutes, this person has more Bacteroides. You feed them both inulin, and you get a completely different profile of short chain fatty acids out the other side. For the same exact amount of inulin put into the system, you get a different output. So you feed two people banana, and you get different responses to that same banana, which really argues for, we have to take the microbiome into account when thinking about nutrition, diet, and health, because it's very likely that we might need personalized approaches to address some of these complex diseases like, irritable bowel syndrome and so on.
Right. So talking about complex diseases, have you thought about the interplay between disease state and microbial diversity or composition in that, so certain complex diseases, and the question comes in related to HIV, and HIV patients are known to have leaky gut. So is it the host disease state driving changes in microbial composition and activity, or vice versa, and what that complex interplay may be to set that equilibrium and the change from homeostasis to managing and helping to manage a disease state?
Yeah, correlation or causation.
Correlation
right?
or causation.
Is the microbiome actually contributing to the etiology of disease, or is it simply a passenger responding to shifts in the body? And this is a question that hasn't been well resolved for many diseases until more recently. A lot of the initial work in the first decade of microbiome research has been kind of the natural history of just seeing what's there, who's there, and what they're doing. In surveys, right? You just have patients that already exist in the world, healthy and sick, and you're comparing the two. In order to get causality, you need to have a few different things, right? You could have maybe a time series that gets you a little closer. If you look at longitudinal samples, you can maybe ask, what came before what? Did the microbiome change before the metabolome and the host changed, or was it the other way around? And then the ultimate way to get causality are experiments, perturbation experiments, or giving someone a chemical and seeing what happens. And so for certain diseases, we have strong causality, right? We know that fecal transplants resolve recurrent C. diff infections in vast majority of people. That's very well established. The causality of disease and microbiome is established in that case. There are specific metabolic deficits in the host that we know can be resolved by probiotic intervention. So phenylketonuria, the inability to break down phenylalanine, genetic disorder in humans. There's been some recent papers showing that you can actually give a probiotic bacterium that can degrade phenylalanine as a treatment for this particular genetic disease. And other diseases, we have strong evidence for causality, like colorectal cancer is a good example, where things like Fusobacterium or toxigenic Bacteroides fragilis, these have been implicated as potential causal forces in carcinogenesis and tumorigenesis, but that hasn't been strictly demonstrated in humans yet. It's been demonstrated in mice, so we know that we can give certain strains of, say, Fusobacterium to a colon cancer model in mice, and you get more colon tumors in that mouse. In humans, we aren't quite there yet. And so for most diseases, that's where we're at, where there's a lot of smoke, there's a lot of evidence for potential in causality, yet we still need to get that next little bit of a step forward to get towards causality.
Yeah. Given the number of times this question's been answered, I think we should address it. Plasma versus serum. Do you expect to see any differences in there? I mean, circulating metabolites or circulating metabolites. We view them as very similar in terms of matrices metabolomically, but Sean, what are your thoughts? Just something to say, because a lot of questions have come in plasma versus serum, to be able to do this kind of analysis.
Well, Greg, I'm afraid I'm out of my depth there, sadly. I consider myself more of a microbe guy, and so that when it comes to the human side of the equation, I'm still an avid student. So I don't think I can answer that question.
So what I would say is that yes. So plasma and serum are tell a very, although there are different subsets of metabolites within the two matrices, they tell a very unique story in terms of while the levels may change between them, they can't directly compare between plasma and serum. Comparing within serum or within plasma samples, they tell a very similar story. So I would say plasma and serum would be similar in this regard for this kind of approach. Sorry, just that question came up a lot. One in terms of gender differences. So you mentioned the androsten derivative and testosterone. Your thoughts on gender differences and gut diversity as it relates to gender-specific diseases and differential profiles of men versus women, especially as they age. What are your thoughts on that, and maybe thinking about studies on that?
Yeah, it's a great question. A lot of prior analyses and prior work I've seen and done on larger human cohorts, I've never really seen a super strong gender signal in the gut microbiome. that doesn't seem to be something that really segregates people all that much in who lives in their gut. However, in this recent paper with linking the blood metabolome to alpha diversity, obviously we saw sex hormones. So that testosterone derivative I spoke about, this was actually found in both men and women, and it was positively correlated with alpha diversity in both men and women. But the sort of average level of that metabolite was higher in men than it was in women. So there was this consistent relationship with this sex hormone that was present in both men and women, even though it was more abundant in men. There was I think 2 other testosterone derivatives in that 40 metabolite panel, and there was 1 progesterone derivative, which may be more specific to women. So I think in some of these sex hormone microbiome interactions, there could be interesting signal to dig into, and we haven't done a huge amount of work in that yet. I'll also kind of call back to the aging microbiome research that Tom Wolmansky is working on currently, where in that study, he found this association between this uniqueness variable and healthy aging. He also found that in women, as they age, that they tend to get more unique faster than men do, which sort of also correlates with women living longer than men and aging more healthily than men in general, but we don't quite understand that interaction yet. So there's a few interesting little stories that we can point to, but by and large, gender doesn't seem to be driving huge differences in the composition of the gut microbiome.
Great, good to know. So as a former liver researcher and GI researcher, we always bandied the term about a leaky gut, and it's still, in my view, a poorly defined enterprise clinically, and certainly not well understood in terms of how it may impact complex diseases and other diseases. Have you thought about applying your technology and your approach to help define quantifiable metrics of what a leaky gut is and what those metabolites could be that could resolve or provide some sort of metric for that?
I think that's like a really worthwhile research question to dig into. We haven't specifically targeted that at the moment, but I think it's on our minds for sure. Yeah. And we have a hint at what might be going on with this alpha diversity paper, in that we see in obese folks the breakdown in the predictive nature of our model, suggesting that there's like a different sort of interrelationship between the blood metabolome and the microbiome in folks that have higher levels of inflammation and obesity, which likely means they also probably have more permeability in their gut. There's probably a signature of permeability in the blood metabolome. I would guess that one might be able to kind of pull that out and maybe more quantitatively and more concretely define what that is, what that looks like. There's a lot of interesting hints in the literature, but I'm super recently been thinking a lot about neurodegenerative disorders, because we have collaborations with Nathan Price's lab on that. And, for a lot of these neurodegenerative disorders, you see constipation or chronic constipation often preceding neurodegeneration for decades or years. People having a lot of constipation. Constipation can kind of cause leakiness or cause disruption in the walls of the colon, more permeability. And, there's some cool work out of Nathan Price's group. Corey Funk has shown, and Priyanka, one of the grad students working with him, have shown that secondary bile acids are kind of enriched in the brains of Alzheimer's patients who have had autopsies. These bacterially derived metabolites are kind of accumulating and increasing in abundance in the brain. And it's curious, right? They're definitely not produced there, right? They're produced in the gut, and they're somehow getting into the brain. But why they're more abundant in neurodegenerative cases versus not, perhaps points towards more leakiness, right? That there's perhaps more inflammation at the blood-brain barrier or at the wall of the colon, and you're just getting more of these bacterial metabolites that perhaps shouldn't be getting to different places in the body. They're traveling there because of this increased leakiness or this reduced ability of the body to filter these microbial metabolites. I think it's a super fascinating question, and there's a lot more work that needs to be done to look into it.
Yeah, Yeah, fascinating, really fascinating stuff. And yeah, you can imagine how this could be applied to understand the role of the gut barrier, and how that's going to influence all systemic illnesses. And it's really exciting. Wouldn't be anything if we didn't talk about the pandemic and everything that has made us work remotely in this regard right now. So COVID-19 and the current infection profile or situation. Any thoughts on microbiome, the role of your approach in gut diversity for COVID responsiveness, the stratification of severity, thinking about that for this particular pandemic?
Yes. There are a lot of folks thinking about this. A lot of labs are thinking, should I pivot my entire research program to studying COVID? We haven't really done that in my group. We're kind of focusing on what we know how to do well, and letting the people who are already good at virus research kind of work on that. There's actually Jim Heath, the president of our institute, has begun a clinical trial on COVID, kind of doing a deep dive into the immune phenotypes of COVID patients who have severe and non-severe disease symptoms. We've done some speaking with Jim on, if he needs our help looking into the microbiome of those patients, we're happy to help with that. I think it's just difficult to get samples from patients, and everyone's exhausted and overwhelmed already. And so the microbiome wasn't a priority. There perhaps are some samples that will be available soon that we'll probably sequence and look at. And I know that Nathan is working with Rob Knight's group down at UCSD to perhaps sample and sequence convalescent patients from this same trial. And then maybe we'll get a picture of what's going on there. It's almost certain that we'll see some kind of signature there. Whether or not it can be used to stratify patients or predict disease severity is to be seen. There was a slightly tantalizing preprint out of China a few weeks ago that looked at kind of the microbiome and COVID. I give you my quick high-level view on that. The paper wasn't perfect. They actually didn't sample the microbiomes of COVID-infected patients. They had taken sort of data from a previous study where they had sampled like 30-some COVID patients and looked at blood inflammatory markers of those patients, and had like a prediction index that they came up with for disease severity. So certain blood proteins that seem to be predictive of disease severity versus less severe disease. And then they took a healthy population of people and applied that proteomic prediction index to that population, so to predict everyone's potential severity, and then associated the microbiome with that. And so they found some signal there, although it's a sort of convoluted analysis. And as far as I've seen, that's the major paper that tried to look at this that I've seen out there. There's probably been a couple others that have come out. It's just a fire hose of preprints these days. Yeah, I mean, it's something we'd like to look at. It's almost certain that we'll see a signal. We know that COVID is infecting the lining of the gut. In fact, goblet cells, which are the cells that produce mucus in the gut, they actually produce a ton of ACE2, which is the protein that COVID binds to enter into the cell. So it's almost certain that these mucus-producing cells are the ones getting targeted, and at least initially where the infection kind of invades the lining of the gut. Jim Heath's lab actually has some interesting staining data from some autopsy patients that unfortunately passed away from COVID, where we're seeing these white hot regions of the virus in the lining of the gut. And looks like they're infecting immune structures. So I think there's a lot of really cool research to be done there. We're just limited in what samples we can get, and I don't want to overwhelm the system from focusing on what should be focused on, which is probably the immune response at this point in time.
Yeah, agreed. No, thanks for that perspective. Staying on virus for a little bit, have you thought about applying phage display, or looking at phage biology and its impact on gut composition and how that may impact different disease states? So the ability of phages to lyse specific bacteria and how that would impact the technology and the approach you're utilizing. Has that been thought of at all?
We're not doing a ton of phage work ourselves. It's a really interesting line of research. Anytime you sequence a metagenome, which is sort of just randomly sequencing all the short fragments of DNA from the gut, you see phage. You can detect the phage that are there, whether or not these are prophage that are integrated into the genome, or these are actual virions flying around infecting cells. We don't know that unless we do special types of sampling where we enrich for virions and so on. So we have data to kind of ask questions about phage in the gut, but we're not actively doing experiments and researching that at the moment. I do find that the field is really, really interesting. There's this whole line of Soviet era research into phage therapy. So, you know, pre-antibiotics, the Soviets were developing bacteriophage as treatments for bacterial infections. And, you know, it was working. It's an approach that can work. And that research sort of fell apart and got buried in history after antibiotics became super popular, because it seemed like it wasn't necessary anymore. But recently, a lot of researchers have kind of resurrected that research. And, you know, there's a few really poignant stories that have been in the news in the past few years where patients with, you know, antimicrobial resistant infections that are on their last legs and have no hope of beating a disease have gotten these experimental phage therapies and have pulled out of it. And the phage therapy has actually worked to kill these antibiotic resistant organisms. So I think it's the use of phage in therapeutics is a huge new weapon in our arsenal that we should be developing and pursuing. And then phage in and of themselves are just fascinating to study because they're these interesting top-down controls on the ecology of the system. They definitely affect the physiology and the metabolism of the microbiota, and shape its structure and composition, and facilitate horizontal gene transfer. So the movement of genes between different kind of unrelated taxa.
Interesting. I had never thought about that. And I think it kind of, to round it out, because it represents a number of questions as well, I think people are sitting on a lot of stool samples that they'd like to maybe implement your approach, and they want to know stool samples, the utility of stool samples for this kind of approach for predicting alpha diversity and gut diversity relative to circulating metabolites. Have you looked at the comparison between the two? Is one better than another? I think there seems to be an understanding in the community, some sort of bias that maybe stool would be better for predicting gut diversity. But what are your thoughts on that?
Well, yeah, it's almost by definition, stool is better, right? Because that is the standard by which we are judging everything. So, you know, the stool calculated alpha diversity is the alpha diversity metric that we're like judging how well the metabolome can predict it, right? Obviously, you can much more accurately calculate diversity of the gut microbiome in stool through stool. That is just by definition true. We just think it's interesting that you can actually see a blurred reflection of that in the metabolites that happen to be bathing every organ in our bodies, right? That there's this intimate connection between our microbiota and our bodies, as if it's like an endocrine organ itself producing things that are affecting our phenotypes. That's where we're coming at it from. Not as like a way of, you know, gold standard method for predicting alpha diversity. It's more of a scientific question and tool for understanding that interrelationship between the body and the ecology of our guts.
Understood. And where do things stand for imputing colonic luminal metabolites in the blood? So I think this goes back to kind of that the separation. Can there be separation for the different structures of the gut? I think we kind of already talked about that and the contribution for the various structures.
Yeah, we try to get at that with modeling to some extent to predict, you know, what the luminal metabolic content would be. You can do invasive sampling, right? If you have a doc who can do like a colonoscopy, perhaps you can get some samples of the lumen. But in humans, it's very difficult to get those samples.
Yeah. All right. Well, because everything else kind of fell under the questions we already talked about. So I think that sums up the Q&A session, and I'm glad you left this much time for the question and answer session, because we essentially used all our time up on a really robust Q&A session. So I would thank everyone for their involvement. I would thank Sean for his time and his deep thoughts on this and his expertise in this area. Obviously, I think we're just scratching the surface here of what really can be lead to powerful understanding of functional outputs of gut activity and microbial activity that influence health and wellness and transition to disease. Any closing thoughts, Sean?
No, just thanks, Greg and Carolyn and Metabolon for organizing this event. It's great for us to be able to talk about our research, and I'm glad that so many folks are excited about the microbiome.
All right. Thanks, Sean.
Well, thank you. Thanks, John. Thanks, Greg. And thank you, everyone, for attending.
Bye, everybody.
Bye, everyone.